I am Erin Wright, and I attend the University of Delaware as a plant science and biological sciences double major. I spent this summer working in UD's Kohler lab with Dr. Alyssa Kohler through the Envision program. I am determining the severity of pod and stem blight disease in soybeans caused by the fungus Diaporthy longicola. Until recently, it was not a focus in this region because most soybean disease was believed to be caused by different Diaporthy species. Diaporthy species cause seed decay in soybeans and reduce yield. Recent UD Master's Program graduate Alexandra Kessler found that the majority of fungal soybean diseases in and around southern Delaware were caused by Diaporthy longicola, and the disease was more severe than that caused by other Diaporthy species. I am determining if Diaporthy longicola actually causes more severe disease or if she happened to pick an aggressive isolate for her research. If Diaporthy longicola is actually more severe, producers will have to change how they grow soybeans in order to prevent it from occurring. 2 cultivars were selected and planted in early June: Hawkeye, which is known to be susceptible to Diaporthy diseases, and P41T07E, a commercial variety that is resistant to Diaporthy diseases. By late June, all the plants were going well. There was a lot of downtime while waiting for the plants to be large enough to inoculate, so I was given the opportunity to assist Dr. Kohler and her graduate students with their research and help around the lab. This included many different tasks such as making media and amendments, preparing and planting field trials, maintaining fungal cultures, and even PCR. When the soybeans were almost the right size, 12 isolates from Alexandra's collection of samples were chosen. 10 were Diaporthy longicola, and the other 2 were Diaporthy aspelathy and Diaporthy eucherae. Parts of old fungal cultures were transferred onto new plates with toothpicks on them. The plates were incubated until the fungus covered them. The soybeans were inoculated 29 days after planting. An incision was made through the stem halfway between the first and second internodes. Then a toothpick was inserted into the wound and the area was sealed with Vaseline. 2 groups from each cultivar were not inoculated. These are the controls, and the results of the inoculated plants will be compared to them. After inoculation, the plants will be observed for signs of illness. This includes, but is not limited to, wilting, chlorosis and necrosis, and lesions that will form on the stem. Observations will continue throughout the summer until 45 days after inoculation, when the full severity of disease has been determined.